Good afternoon, everyone, and thanks for coming to the Wilson Center to talk about our new report about the do-it-yourself biology community. For those watching on the web, we are going to open this up to a discussion after we sort of discuss the report. And if you would like to send in questions, you can send them to us via Twitter. And our Twitter handle is @SynBioProject, and that's SYNBIOPROJECT. And I'll announce that again later when we get to the question and answer period. So the way that we're going to do this today is Dan Grushkin, who is the lead author on this report, is going to go over the report in detail. And then after that, we're going to have comments from Jason Bobe, the co-founder of DIYbio.org. And then we're going to hear from Ted Myatt, who is one of the biosafety experts that's part of the Ask a Biosafety Expert program, to give you his thoughts on that program and the DIYbio community in general. And then if we can twist his arm, we're going to have Special Agent Nathan Head give some perspectives from the FBI on the DIY community. So with that, I'm going to turn it over to Dan Grushkin.
So thank you, everyone, for coming. What's that?
Just talk into there for the web folks.
Thank you, web folks, for showing up as well. So I'm going to just give you a quick explanation of the report and some of the findings that we had. So here's a short introduction on DIYbio for those of you who don't know. Some of these are probably familiar to you. The first thing I'll point you to is the Carlson curves, which shows the dropping prices of synthesizing synthesizing and sequencing DNA, reading and writing DNA, and how precipitously they've been falling over the last couple of years. That's Rob Carlson right next to him. Rob Carlson's probably one of the original do-it-yourselfers. He started the lab in his garage, which I'll show you a little later. Below, you'll see Jason over here and Mackenzie Cowell. They started a website called DIYbio.org, which was sort of a forum for people to get together and collaborate on DIY projects. Jason, do you want to give a little background on it?
So this was, it started in about 2008 and actually had a really local basis to begin with getting people together in Boston who were interested in talking about and doing biology more as a hobby than as just a profession. And my interest grew out of working in the church lab at Harvard Medical School and seeing him invent DNA sequencing technologies that looked like a washing machine. They basically hacked a microscope and then made a device that looked like a washing machine. And I said, I would love to have one of those to do my own sequencing projects. And I'm going to go look for other people in Boston who also would like to work with me on DNA sequencing projects. And that was my initial interest in going out and starting a DIYbio group with Mac, who came from the International Genetically Engineered Machines competition. And so we started groups and started talking about it and blogging about it. And there were lots of interest around the world immediately, and people started setting up groups in their own cities. And Dan will talk about sort of the contours of the community and how they've changed in the past 5 years now.
So immediately upon the rise of DIYbio, we started getting media reports about DIYbio, and that's a lot about what this report is about. The myths that have kind of arisen with these articles, and actually trying to basically find the truth within these myths, or whether there is any truth to these myths. So we started getting articles like In Attics and Closets, Biohackers Discover Their Inner Frankenstein, Biotech in the Basement, The Geneticists in the Garage, all about how basically people are doing biotech in unconventional, untraditional spaces, but they were tinged with a fear, right? The Frankenstein fear. And here are some of the negative framings that we got around amateur biology. So I'll just read off a couple of them. Welcome to the age of synthesized life, built from scratch. Soon it may be cheap and simple for a teen hacker to do it, or a terrorist. I'll give you another one. With DNA hacking far more widespread, what if your friendly local terrorist decided to take up the hobby? So you see that basically when lay people want to get involved in this, in actually working with biotechnology, we get this fear factor that comes along with it. Here was something interesting in my experience. One of the first conferences I went to, this is actually a slide from someone else. It was someone from HHS who showed the slide and said, "Here's a case of DIYbio," and they pointed to this to this guy who was making anthrax in his basement in order to poison the U.S. Army troops during World War I. Actually, their horses. So that was sort of a wake-up call to me that the message in the media had spread to the policy community, and I was sitting in the audience and watching people talk about the DIY community as a serious threat, or in this case, a traitor of sorts. Let me tell you a little about the survey. Essentially, we went out and polled the message board, the DIYbio.org message board, but we also went to individual community labs and asked the directors of each of these labs to get their members to also answer the poll. And it was a poll of, I think, 27 questions. The incentive for them to actually answer was that one of them would win a open PCR, which is one of these. One of the findings we found, and I think this is pretty startling, is that the gross majority of people had started, or the gross majority of respondents had started in the last year, even though the organization had been around since 2008. That says that it's growing incredibly rapidly. I think basically more people joined in the last year than in the 4 previous ones, which says to me that maybe next year that number will increase again, and there'll be a whole fresh batch of do-it-yourselfers out in the world, an expanding movement. The other thing that was interesting was that the people who were actually doing the experiments, right, the members themselves, they had some background in biology. Most of them had studied it, or the majority of them had studied it in college to some degree. And then you see that basically it drops off. So the next group would be the high school, people who studied in high school who have obviously very little experience in biotechnology. The myths that arise from the media are as follows. One is DIYers work anonymously and solitarily. We saw that in the previous slides where this is Kay All. She actually had a closet lab. And so the first round of media was all about that other people might have closet labs or doing things we don't know about. The other one is Rob Carlson. This is his garage lab. I love this graphic. This is from Nature. And it shows basically how cheap it is to make your own home lab. The reality was that soon after this movement arose, something called what we call community labs basically became the majority, the place where the majority of actual DIYers work. And so this is the first one. This is GenSpace in Brooklyn in June 2002. That's actually my back. I was laying tiles that day. And basically it became one of the first community labs. I'm just going to show you a video of what that lab looks like. So from this mess that we encountered to what it actually became. Let me just exit out of here. That's it.
GenSpace is one of the world's first community biotech labs. We're located in Brooklyn, New York City, 33 Flatbush Avenue. There's more and more people getting into doing biotechnology experiments outside of the traditional venues of academia and industry. The technology is becoming more and more accessible. Information is much more easily obtainable, and at the same time, it's becoming more and more relevant in everybody's lives.
This is the smaller of our labs, and it's filled with equipment that's either bought off eBay or donated or on loan. We have a spectrophotometer, a PCR machine to copy DNA, an incubator shaker. These are mouse cells. You can't see them because they're microscopic, but they're in there, trust me.
Space is really at a premium. That's the hard thing to get at when you want to do experiments. Madame Curie, discoverer of radium. When she was working in France, she was complaining that she had no space to actually do this groundbreaking work, and all she needed essentially was just a shed. You can't order this type of equipment and these reagents and do this stuff in your bedroom.
We can do gel electrophoresis. We have 2 microcentrifuges. This is a bacterial incubator, and you can see we've got some very pretty red bacteria. This is E. coli that's expressing a red fluorescent protein. See?
Most labs, though, focus on one thing, one disease model, one protein, one pathway. Here we have a number of individuals working essentially shoulder to shoulder, and all of these people have different skill sets. We have a number of artists. We have somebody who comes from a background of patent law. We have engineers. Because we're so interdisciplinary by nature, we're essentially increasing the probability that somebody's going to come up with something great. We do not take any intellectual property. Everything that you do here, just like in a hackerspace, it's yours. So people are free to invent here without any worry that anybody's going to steal their idea or their invention. There's a lot of things amateurs, in the truest sense of the word, can do to contribute to science, and have done. A couple of bicycle mechanics invented the aerospace industry. Gregor Mendel, he was a complete self-taught amateur. We've seen it happen with the PC revolution. It sort of harkens back to traditional American craftsmanship, where people would start essentially industries out of their garage workshop.
So soon after GenSpace opened, we had BioCurious in San Francisco, and it kept on growing from there. At this point, there are around 14 or 15 community labs. Here's Biospace in Canada, Berlin, Indonesia. Hackteria's basically a one-man shop that travels around the world. Prague, Manchester, Paris, and more. I didn't take them all. If you look at actually what people said, where they worked, you'll see that the majority of them or most of them either worked in a community lab, a hackerspace, or an academic, corporate, or government lab, which says to me, or actually, if you look at the numbers, it's something like 92% of them, of people actually work in group spaces, which is, I think, for the biosecurity world, a positive sign. It says that people are collaborating with each other, they're available. You don't really have this guy working in a cave, the guy working in the closet who's not paying attention. He's surrounded by a community. Interestingly, you'll see that so people work in multiple different spaces. So someone who might work in a community lab one day might have a home lab that they take their experiments to, or a hacker space that they go to. There's a lot of moving back and forth. Only 8% of people actually worked exclusively at home, a very small number. But it seems to me that community labs have really taken in most of the people who consider themselves do-it-yourselfers. Here's another myth. DIYers are capable of unleashing a deadly epidemic. In the last couple of years, we've seen a lot of reports that suggest this. Probably the most well-known was the New York Times report that said, this is right around the flu controversy about releasing the sequence of the flu. One of the fears that they mention is that amateurs might be the ones who create this new mutant virus, new potential pandemic. Another example is The Atlantic. They had an article called Hacking the President's DNA, which basically was about a bunch of sort of science fiction in a way, how potentially anonymous biohackers or do-it-yourselfers might actually create a virus that would target specifically one person, that person being the president. A little far out there. I'll give you another example, PBS, which I think of as probably the most serious of journalism in the United States. We had a reporter come to GenSpace. We told him what he did. When the report came out on TV, they basically had us juxtaposed with someone who was talking about how one might hack into pacemakers and turn them off, which really had nothing to do with what the DIYers do, but somehow they had been associated with this kind of stuff. Then you have the biocrime report, which was a Wired article about all the dangers associated with regular people having access to biotechnology. The origins of these kind of fears are pretty obvious. I think we all know about the Amerithrax crisis that happened after 9/11. The other thing that I think is interesting is that in 2002, Eckhard Wimmer, a lab scientist, had actually synthesized the polio virus, which showed that it could be done chemically. And I think there was the fear that if someone could do it in the lab under these conditions, you'll see everyone's in special outfits, that it might be able to be done in a do-it-yourself lab. What I found is that when I asked people what they did, these kinds of experiments are just far, far away from what actual do-it-yourselfers are doing. And I'll just give you some numbers. Probably the most interesting is that around half of people said they've genetically engineered bacteria or yeast. And what that means is usually they've taken a plasmid that they've gotten online or through a lab, and they've put it into a bacteria. That doesn't really take much. One of the first experiments you do in a molecular biology lab as an intern, or even now in high school classes, is do a genetic engineering experiment where you put a plasmid into an E. coli and get the E. coli to express GFP, green fluorescent protein. If we were to look at that as an analogy with computing, that would be the equivalent of taking a CD, right, a pre-made program, and installing it on your computer. So only half of people have done that. The other stat I thought was interesting is only less than 20% had synthesized a gene. That would be the equivalent of writing a program. So when we're talking about making designer viruses, or we're talking about engineering the next pandemic, at least from the respondents, it looks like no one's even near that level of sophistication. The majority of people had extracted DNA, which is actually a pretty common experiment that people do in their home labs. Usually it's from a strawberry. They mash up the strawberry. They pull out some DNA. We've actually done it at AAAS with the FBI. It was a pretty fun conference. Another myth, DIYers are incapable of contributing to biotechnology. I think that actually the DIY community has already proven this one false. Here's Nature Biotech basically making the claim. One of the questions I asked when I addressed that myth was, what is a contribution? How do you define a contribution from a do-it-yourself biology community? The way that I separate it is technical scientific achievement, a business achievement, right? So someone started a small business, or contribution to education and public awareness. I think that basically it falls into roughly those categories. Here's an interesting one. So this is also out of GenSpace. Essentially, they raised $7,500 to collect plant samples from the far reaches of Alaska to actually get the DNA sequences of various plants and start sort of an ecology project where they could go back to this place over and over again and measure the way that plant life is changing as the climate changes. Project's not done. The plants were collected. They're now basically once a week at GenSpace. People come and barcode these plants, actually get their DNA sequences for something no one had ever done before. Another great one, this is a small business and a technical achievement. This is Amplino. This is a team of do-it-yourselfers in Amsterdam. They won €40,000 from Vodafone to basically work on this project where they had already have a proof of principle. And basically it's a very cheap diagnostics kit for malaria. As far as I understand, they're going in the next couple of months to Africa, I think Mali, to try to test it out. So a small business has basically risen out of that group. Here's another one. You see the open PCR. This is a very inexpensive, basically a DNA copy machine. A group in San Francisco did this. This is also a small business, and these are not only sold to do-it-yourself groups, but labs actually buy these as well. This I thought was interesting. Here's Jack Andraka. So he won this year's Intel Prize. Essentially, when he was looking for a lab to work on his project, he was basically it's a cancer diagnostics kit that he was looking to do. He couldn't find a lab. He sent out letters to 200 laboratories. Only one said yes. And voila, that lab is what allowed him to win the Endel Prize and make this diagnostics kit. So what it seems to me is that if you give someone the opportunity to get a space, that there is potential for them to do very important work. This is another GenSpacer. This is Carlin. Yeah, he came to GenSpace when he was 15 years old. By the time he was 17, he had created this thing called the OpenFuge, which is a very cheap open source centrifuge that actually is sold out online right now. The kits are sold out online right now. I just wanted to give you a sense of him working. So this is him, I think it was earlier this year, working on the project at GenSpace. You see everyone's working around the table with various equipment. Here he is.
Why are you hiding behind a chair?
There you go. There he is hiding from his experiment, hoping it doesn't fly off at him in the face. But a really interesting technical achievement on his part. So at least I think that these are the beginnings of contributions. Another myth, DIYers risk accidents and environmental release of genetically modified organisms. This is a hot button issue right now. Just to get a sense, in community labs and also I believe in a lot of the hacker spaces, there are rules that the labs impose on their members. One, for example, is no one is allowed to work with pathogens. They adhere to BSL-1 lab standards. They make sure that they dispose of their trash properly. They actually hire out services to collect them. It seems to me, in terms of environmental lease from the community labs, there's a low chance of this happening. Because people work in different spaces, there's a small chance that someone might be transporting something back and forth and might drop it. But even still, because it's BSL-1, it just doesn't seem likely. I think another example, do you want to talk about this one?
So ask a biosafety expert. You know, the biosafety profession typically, for the most part, ends at the perimeter of the institutions with which they work. And as there are new settings like community labs, the Ask a Bio Expert service is a chance to extend that professional reach into these new environments like community labs. And so we have assembled a group of volunteer advisors who are the biosafety experts at many of the major academic centers and biotech institutions to collect questions from DIY community members and then provide responses and biosafety advice and recommendations along with links to open access resources and publish those in a knowledge base. And so we've piloted this program with 3 brave biosafety experts. It took about 2.5 years to put together to go actually get professional liability insurance and those sorts of things and find experts who wanted to try something new. And then, so we piloted it and we just went and I made a presentation to the American Biological Safety Association to ramp up the service. That was a few weeks ago, and we're now onboarding about 12 more biosafety experts to join the service and expand our offerings to the community members and community labs. I don't know if we were going to have Ted jump in, but Ted Myatt, one of the biosafety advisors, is on the phone, and for questions. Or Ted, if you have any comments, you're welcome to jump in now or wait for questions. Is he there?
Are you there, Ted? All right, we'll come back to him a little bit later.
Okay, I mean, I think the main point about this is that if, in terms of biosafety, there's a resource to do-it-yourselfers who are not necessarily working in community labs to find out whether what they're doing is the right thing, and if they do have problems, they have a resource at their fingertips. The one concern I think among or the hot button issue right now is about one project called the Glowing Plant Project. This was kickstarted over the spring. Essentially, a number of do-it-yourselfers put together a Kickstarter saying that they would make a glowing plant, and one of the rewards they would give in return for giving money to actually do this project would be to send out seeds for these plants. And this sort of leapt over a lot of the regulatory frameworks that we have in place with large seed companies like Monsanto. There's some big questions about the project, like how much glow can you really get out of the plant? How's the regulatory body's going to react? The jury's still out on a lot of this. But again, this is an interesting case because it's intentional environmental release. And I know, at least in the DIY community, it's very controversial, as it is outside of that community. Next one, group labs may become unsuspecting havens for bioterrorists. This is, I think, a concern among a lot of the policing agencies, a real concern that someone might infiltrate one of these laboratories and use the equipment to make something that's dangerous. I think there are a number of hurdles for someone who with nefarious intent, and these are them. So we asked the DIYers their feelings about transparency and sharing their work, and you see that most of them are strong believers in transparency. I think the fact that everyone is curious and wants to show their work is one hurdle. I'll give you another hurdle. This is about a code of ethics that came out of the DIY scene in 2011. Jason, you want to take care of this one?
Sure,
too?
yeah. So the thumbnail sketch of this is with the support of the Woodrow Wilson Center. We organized several congresses, one that was held at the London School of Economics, and invited DIY members from groups throughout Europe. And then we held another congress in San Francisco, and it was a all-day workshop to assemble a general aspirational code of ethics. And so each one of them, they worked independently, but came up with codes, I think, that were very similar. And this is not, of course, anything that is binding on any member. There is no overarching organization that everybody belongs to, but it was a conversation. It was a conversation starter for the movement wanting to set the tone of building a vibrant, safe, and productive DIY community. So this is about this. This is look. There's a date there, 2012. I actually wasn't at this meeting. I had my first daughter on May 16th, 2012. Josephine. I was sleepless at that time, but our relationship with the FBI goes back almost to the very beginning of DIY biology, and we've worked with them very closely for years, basically as to educate each other. It is, there were strong feelings by many of the community members of being afraid of the FBI to some extent from the Steve Kurtz case, which sort of set the tone of fear from the authorities to some circumstance. And on the other hand, people in the very early stages, with the sort of heightened climate of awareness and fear around terrorism, there was it was a difficult time to be a DIY biologist, even if all you were doing was extracting DNA from strawberries. So it was very important for the DIY community to educate the FBI and the FBI to learn from us, and for them to tell us sort of what their mission and goals are in national security and their concerns. And I think that has evolved to be really productive. for the most part. There still, I think, are elements of the DIY community who are suspicious of course, of intrusion, but
Yeah. I mean, in terms of the Smith, it's important to note that all of these groups have a relationship with the FBI. So if in fact you were a nefarious character and you were going to try to infiltrate one of these labs, most of the directors of these labs carry the cards of their local FBI WMD director in their wallets. So
actually, just a point about just jumping here real quick, just recalling a scenario about how the ebb and the flow of being part of the DIY community. So there was, in a sense, the role that is played by media and policymakers who are trying to understand what's going on. One of the groups invited the FBI to attend one of their meetups in Baltimore, and a journalist covered this event, and the headline of the blog post was, FBI takes interest in local DIYbio group. Obviously painting a certain sort of picture that set the tone, which was, I think, consistent with a lot of how when people hear about DIY for the first time, for the most part, between 2008 and 2011, that was how they were introduced. Even though it was the relationship underneath was something that was entirely different.
I think that the final thing that's a deterrent for a nefarious character is the fact that most of these labs are group labs. No one's working in isolation. There are lots of people all around them looking at what they're doing, being nosy, asking questions, asking for help, sharing. It seems like it would be a terrible space to try to do something, nefarious. One last myth. DIYers are versus government oversight, which is, I think, a general sense about what the DIY community feels about oversight, feels about transparency. I was surprised by this statistic. So we asked them, should there be government oversight now or in the future? Now, most DIYers said, no, we don't think so. But in the future, the numbers start to even out, and that was really interesting. It should be noted that by oversight, I think that there's a sense that usually that's clamping down. I don't think that's necessarily the case. I think there are carrots, incentives that could be considered oversight as well. That would work too. There was a comment field, and a number of the DIYers sort of tried to explain when that moment should occur, when oversight should change. Some of them thought it should change when all of biotech new laws came into effect, and so they would be considered just like other biotech labs. Others said, well, when a specific technology, for example, I don't know, one person said a DIY printer, a gene printer you can have at home might be the moment when the oversight should change. But I think that the point here is that there's the DIYers are willing to engage in debate. And so this is a great opportunity to bring them in on this debate, because obviously they're not stalwart on this issue. The recommendations were as follows, but I'm going to just focus on one, which is education. And that's basically that education plays a major role in all the DIYbio groups. And I'll leave the other ones for you to read the report. One of the major recommendations in the Presidential Bioethics Commission report that came out in 2010 was the idea of educating the public on synthetic biology and biotechnology. Along with universities and government labs and private foundations, they included grassroots scientific organizations, exactly like DIYbio, to spread the word, basically do public education. And I actually think that the DIY community, without any support from outside sources or from government, has already begun to do this. When we asked people what the main, how they would categorize their projects, what they were working on and why they were working on it, the first thing they said was education. And it was closely followed by personal exploration, which is sort of, studying these things for themselves. But I think it's significant that education was the number one response. And we've already seen that. So here's Natalie Kuldell, an MIT synthetic biology professor teaching at one of these community labs, architecture class, students this is actually a DIY group going out into science fair and teaching young kids. iGEM team, we talked a little bit about iGEM a little while ago. So this is probably one of the first DIY iGEM teams. Actually, this year, iGEM opened up to any DIY team. Oh, sorry, next year, the year coming up. So that'll be an interesting phenomenon. Here's a mosaic project, an actual interactive project at South by Southwest 2012. I don't know if you can tell what that is. It's actually a mosaic unicorn made out of GFP and RFP E. coli. Kind of funny. Here's a great public outreach. This is an artist. Her name is Heather Dewey Hagborg, and she's interested in the questions of privacy around DNA. And so we obviously shed DNA wherever we go. We leave them in our cigarette butts, our chewing gum, our hair follicles that we leave all over the street. She actually goes and picks those things up and tries to figure out what the person who left them looks like by sequencing their DNA and putting in the characteristics of that person into a sophisticated computer algorithm, and then print them out into these really interesting busts. So that's a very short summary of what's written in the report. Thank you all for coming. I'm glad you're all here. Pass it to you.
Yeah, so thanks, Dan. So I want to reach out to Ted, who's back on the phone, and hopefully, Ted, you can hear us. And if you could maybe give your thoughts on the community, and then also the Ask a Biosafety Expert program, and what drew you to that program, and maybe some of the types of questions that come in through that service.
Sure. So can you guys hear me?
Yes.
All right. There's a little bit of a delay, so if I talked over you just now, I'm sorry about that. So I am a biosafety officer, or have been a biosafety officer in the past, and was approached by Jason back when I was working up in Boston to participate in this program. And so it's been, I think, really just as educational for the biosafety community and the biosafety officers that have participated in this program as it has, hopefully, for the folks that we're providing some guidance for. I don't think we necessarily knew what to expect when the questions started to come in, and it's been a bit of an interesting time for us, I think, in figuring out how to respond to some of the questions, because while I think a lot of the research, like Dan was describing, is not high-level research in terms of safety risks, dealing with folks that aren't in a traditional institution where there are institutional resources available for, chemical disposal and the things like that, has provided some interesting things we've had to think through on how to best answer the questions and provide good recommendations. So, in general, the questions have been very, I don't know what the right word is, benign. They've been very good questions for the most part, in terms of, really wanting to conduct the research safely. A lot of the questions have centered around actually chemical safety as opposed to biological safety, I think. How to, can we dispose of this chemical down the drain? Is it okay to put bleach down the drain? I think they were asking questions that if they, people would regularly be doing that sort of thing in their home, but they wanted to do what is absolutely, what's the best practice in terms of laboratories. So in general, I think the questions have been really, interesting questions, but questions that do have answers. And from what we've seen anyway, from the folks asking questions, we haven't seen really anything that sort of has raised an eyebrow or anything like that in our community as research that is not appropriate for the facilities that they're conducting that research in. I was just going to mention one thing about the government oversight. That's always been an interesting question about the slide about government oversight, if that's needed or not. And I think it's important to point out that the document that the institutions that conduct this sort of research regularly follow is the NIH guidelines for research involving recombinant or synthetic DNA research. And that document really only applies to institutions receiving grants from the NIH. So there are plenty of private biotech companies right now that don't need to comply with sort of the most basic document that we use in our industry to ensure safety. So not to say that they aren't doing things safely, but they don't have to follow that document. So they even right now they have little federal oversight for a lot of the research that's going on. So that may change down the road, but it's an interesting thing to think about in terms of additional government oversight for community labs and the like, if that additional oversight isn't also required for or agencies that or institutions that don't receive NIH funds to conduct some of this research.
All right, thanks. So Ted, if you could maybe just hang on, because there might be some questions for you in a few minutes. I want to see if Nathan, if you want to, you can come up here, or we can bring you a mic. It's whatever you're most comfortable with. Sure. To talk about the FBI's outreach program and your experience with the community.
Sure. Oh, you're fine. I just slowly escaped the light. So my name's Nathan Head. I'm a supervisory special agent with the FBI. To keep it short, I was kind of on the leading edge of the FBI's engagement with the do-it-yourself biology community when I was assigned to the New York office and engaged with Dan for the first time in New York with my fellow WMD coordinator, Pat O'Brien. Needless to say that the sea change has happened over the last 4 years. The tide has shifted. The feelings from the community have slowly changed, as you made reference to. A lot of what the FBI is interested in is of course protecting the U.S. interests against terrorist incidents. We do that from my perspective. We're involved in a proactive and a preventative mission to address biological threats. And so we engage and conduct outreach with various areas of the sciences to include academia, industry, first responders, public health, but that also includes entities like amateur biology. There is a perception that started very early about why it is that we were doing what we were doing. But we do this type of outreach in a lot of different settings, trying to sensitize against the potential for someone to be exploited and their circumstances to be used against them. And so we talk with the community about the possibility of their knowledge, expertise, equipment to potentially be used either by an outside actor or maybe someone that's inside. We talk a lot about insider threat with other types of biological laboratories. But the community has been very receptive to that. We have representatives across the United States, our WMD coordinators, who are in each of our 56 field offices. And they maintain relationships with area community laboratories and DIYbio groups that sensitize that message. We are very sensitive to the understanding that science is a very good thing. I'm a scientist myself. A lot of my colleagues are scientists that do this type of engagement. We understand the clear benefit to this. Our actions are not to impede, however, to help guide through maybe this path to address these concerns, because there is a situation where these concerns might come up. It's better to be prepared for those ahead of time and address those with a full-fledged breadth of knowledge rather than have to react to those and have maybe some detrimental action come as a result of not being prepared to address this. And then maybe some other action is taken, either regulatory or governmentally, to address an issue that they see that is a void. So I personally commend the report. I believe the report captures especially the myth that involves engagement with law enforcement. It's been something that I've been intimately involved in the last couple of years. But I think through a lot of the efforts, through a multitude of members of the community, you three here, and frankly, individuals here in the interagency in DC, and as well as our FBI colleagues throughout the country, the engagement has been fruitful, and we've been very excited to see DIYbio amateur biology grow in the way it has, and as well as its expansion internationally. Thank you.
All right, thanks. So, Jason, do you have any other comments that you wanted to add?
I don't think so.
All right, so what I'd like to do is open it up to questions and to have a broader discussions. For those watching on the web, if you'd like to send in a question, you can tweet us that, and our handle is @SynBioProject. So just send us a tweet with your question, and we'll try to get to them. Yeah, Jerry? Oh, wait, just wait for the mic so people online can hear you. Sorry.
I'm Jerry Epstein with the Department of Homeland Security. I had a question on the Ask the Biosafety Expert.
Yeah.
Jason, you had mentioned something about getting liability insurance, which I was glad to hear because I hate that we live in a world where doing a public service exposes you to horrible possible consequences. So I'm glad you've done that. I wonder who's paying for that. How's that supported?
That is through grant funding.
And so for some number of years, you've got it. And then
So it's the same policy that an institution would get, a professional liability policy, an institution would get for a biosafety professional. But the problem is, or the reason why we would need that, is because a biosafety expert's insurance that they get at their day job only extends to the perimeter and for the business held on premise, basically. And so we have a policy which covers their work through DIYbio.org, which is a nonprofit. And that was in the early stages of setting up the program. It was, I naively thought that, great, let's do this. Let's get some biosafety experts. Let's launch a website, and we should be up and running in a few weeks. And not so. So it actually took 2 years to actually find insurance and to talk with insurance companies. It's kind of an opaque industry to go get, and the type of service they were offering was of a different nature than all of the check boxes on all of the forms, and led me initially. It's a long story, but I went to Lloyd's, who will insure anything, Greta Garbo's legs and $1 million half-court shots, and then that ultimately fell through, and I ended up with basically a pretty standard policy that is the same type of insurance that an institution would get. So yeah, it turned out to be a prerequisite for biosafety experts to practice, just like a physician, a medical doctor would have a professional liability insurance. And it also, I think there's an analogy here to, in some ways, with Doctors Without Borders, in that there are physicians feel compelled, feel like an obligation to extend their expertise to people who don't have access to healthcare. And I think the biosafety professional at this point in history, it's kind of the same situation. And just having been at ABSA, I can tell you that there was a pretty obvious divide generationally, that some of the up and coming biosafety experts who were just in the process of doing postdocs and getting their training and going on to into the business were really excited by this, whereas older biosafety experts were at first terrified. They had never heard of this concept of DIYbio and community labs, and so that was kind of palpable. But that's not totally the case. We have very senior biosafety experts who have over 10 years of experience, several of them who feel, I think, that there is an obligation to get their expertise out there in the hands of people who need this information. And we have run into a variety of challenges beyond insurance, too, which is that a lot of this, of the expertise and references and source material that would be readily available in an institution, is behind paywalls, that if we want to create a knowledge base, and not just give a recommendation, but give a recommendation and provide sources so that other people can go review the actual source material and learn more about it. some of those things are just inaccessible. And so, we have to respond in ways that only point to open access resources, and that's sort of the most useful way to proceed. And so we are performing this service in a way that has a variety of challenges. So it's taken quite a while to work out how the service is going to work. But we're at an exciting time where we've got the basic protocol and all the pieces in place. We're going to start working on the model, and there's a bunch of other things that could be really interesting too. Taking a page, just some ideas that could be really helpful out of the page of the playbook of reality TV and remaking your home. It's a real challenge to work with clients who you can't see their lab. And so we need to add the ability to get photographs and videos of bench spaces to understand the context with which people are working. Doing sort of safety makeovers in some sense there's one concept of a sort of spin-off service of this. Anyways, so it's going to be, I think, a really fun couple of years putting together an effective service. And I think it's also a fun opportunity for the biosafety experts to think about their work in new ways. And so I think that's why there's a lot of excitement from some of the younger people who are just starting in their profession.
Peter?
Thanks. That was fascinating and very interesting. I'm going to preface this by saying I fundamentally agree with absolutely everything you said. You are sort of asking a question, should the public be at ease? And you're reassuring the people that the vast majority of practitioners that responded to you want to do everything transparently and want to do everything right. Some of us, like Nathan, are kind of paid to exercise reasonable suspicion. And in my case, we speculate on consequence management. And this may be in the report, which just came out today, so I haven't read it. But it seems to me that this all depends on how you define DIY bio people, as the guild, as you've created it and it's evolved, or people who work alone or perhaps in small groups in independent research on biology. And if so, there's a possibility for some of this to be misleading as it was based on respondents. It only occurs to me that anybody who has malintent would have every incentive to either be untruthful or to not reply. If somebody wanted to evade the community lab restrictions, they wouldn't join a community lab. The equipment's broadly available. So essentially, do your conclusions depend in large measure on how you define DIYbio, and is that definition clear in the report?
So I have two responses to that. The first is basically it's self-identifying DIYers who responded. If you're someone with a nefarious individual and you're obviously not going to respond to the report, I also don't think you would be considered part of the community. Right? At that point, you're now kind of hovering into bioterrorist realm. And that sort of leaves our little enclosure and enters into Nathan's. What would you say?
Yeah, I mean, I think I agree with that. I mean, I think the way that the community is set up, particularly if you look at the codes of conduct, which when Jason and I did that, to be honest with you, we had no idea whether that was going to work. And what was interesting to us is that those two coding workshops, which one was done in Europe and the other which was done in the US, we didn't share the results from those between the two groups. And if you look at those codes, they're almost identical. And so that sort of showed to us that the community that's self-identifying as a DIY community actually is in agreement in how they're going to operate, and whether that's with transparency and safety. And so I think when you start talking about these, nefarious people, we're talking about sort of criminal activities, which falls way outside of the DIY community, and I think it should always be talked about as such. I think a lot of the problems with these press reports, which was sort of the impetus was this, is that they were getting lumped together, whereas someone who was recreating a flu virus to be released out into a community is is doing a criminal activity, and that should be treated as such and talked about as such, and not be lumped together with the DIY community.
It would be like if you were a chemist, and every time that you started to have a conversation about your work, somebody would treat you as a meth lab maker, or somebody who the conversation always begins with, well, what about meth labs? I think this was one of the most challenging things about DIYbio and participating in it, and knowing that there are and will be for many decades all kinds of open questions and issues related to decentralized access to biotechnology, generally speaking, but to create a culture that is transparent and safe, that I think it's important that people talk about what they're doing and discuss it without fear of being treated like a criminal. In the early stages of our engagement with the FBI, the incentives of those people who were open to actually talking with the FBI, it was to prevent one of the slides in a presentation that I gave very early on to the FBI was a picture of a DIYbio lab and a picture of a meth lab, and it says, can you tell me which one is a meth lab and which is a DIY lab? Out of concern for simply being lumped together as a criminal enterprise.
Let me add to that. It just seems to me that we're all stuck on this safety bioterrorism question when we're missing a number of the opportunities that the growth of this community presents. Here's a community that is about open engagement with the public about science, which is something as a country we desperately need. And so rather than continuing the conversation about dangers, I get the sense that it's time to start thinking about the opportunities that this community presents. And those are education and innovation and basically bringing fun into science in a way that we haven't seen before.
Yeah, JD?
Yeah, it's a very interesting report. I want to ask, I guess, Jason the question because he's the one closest to George Church. George Church and Drew Indy have both called for licensing of DIY bios. It seems that the Cambridge would be, an ideal place to write that regulation and to require that licensing. I've suggested that you could take tattoo parlor licenses, change them just a little bit, and they'd fit very well for the DIYbio community. Have you started doing drafts of such licenses? Have you gone to Cambridge or the state of Massachusetts, anywhere, to say this is what we think this licensing regime ought to look like?
Short answer is no. I have actually spoken with George about that, and I can't speak on behalf of George, obviously, but as somebody who he's a technologist, and he is very excited about people using his technology and creating great things. But I think he's also very much a pragmatist in the sense that he feels an obligation to think about schemes that allow widespread use, but also try to mitigate risks. And he always uses the analogy of, if we're going to have laws and regulations of these technologies that or we need to have speed limits. He uses, that if you, and we even have that a license could be totally easy to get, and it could cost $1, and just as easy as it is to get a fishing or hunting license, and we have licenses for all sorts of things, and it doesn't have to be something which is exclusionary by definition, but it could be totally inclusionary.
I actually don't just casually suggest the tattoo
Yeah.
parlor licensing. It's way more than a hunting license. It's even more than a driving license.
Yeah.
Requires a skill set and supervision. If you're not going to follow through on doing that, I would urge you, and I'll tell Dr. Church and Drew Indy, to drop that from the stick, because it's just a red herring if you're advocating for it and not doing anything to implement it.
Let me comment on that. I think that it's something to consider, but I also think that the technology that people are actually using has to get to a certain place for us to consider it. And I think it's probably something that should be discussed, including policymakers and DIYers, about when a benchmark, an appropriate benchmark should come in the technology or in the way that DIYers are using the technology for them actually to implement a licensing platform. At least from what, from my survey, from looking at what people are doing, it just doesn't seem appropriate at this time. That's my perspective.
And also, JD, I mean, I hear what you're saying, but I'd have to push back on your last comment of saying, don't you think that to generalize your last point, is it not okay to start to discuss regulatory options without needing to talk and do at the same time? Or, I mean, I think this is an ongoing discussion, like the Presidential Commission of Prudent Vigilance, that it's something that we watch how technologies are being used and figure out what the optimal approaches are. And I don't know, I mean, it's something you have to have a conversation with George and Drew about whether or not it is the right time or there is a good model and there's good reason to do it. All of these things take energy and effort, and there are sort of endless things that people could be doing in the world, and allocating resources appropriately to things that actually need them, to touch on Dan's point, I think is important. So, it's not an infinite resource world that we live in, particularly for the government or government oversight. It's pretty resource poor. So I would love to see a program that, or resources used in effective manners. I'm not sure that in the DIY community that there is reason right now to invest heavily in a licensing regime. I think it's good to talk about it and to move the ball along. And
Well, I mean, it could put into practice something that you say is already a practice, which is you're only working on BSL-1 organisms. That would, it could make an ordinance that you can't in a DIY lab work even on a organism that's 2. It could put in place, yes, you can work on E. coli, but you can't work to try to engineer E. coli to produce Shiga toxin. Mm-hmm. There are some ways to think about it, and new regulations happen mostly because of weird accidents. While a community lab is a great thing, you wouldn't want to have 30 people exposed to what someone developed, in their lab that they thought was safe. So, figuring out how you do that. Even tattoo parlors have problems with that. Now that nano chemical inks are coming, there's always something new, but it's not a reason not to figure out how we regulate it. The President's Commission was wrong on some things. They said the coordinated framework would work, and the first thing that comes out for plants, coordinated framework, we knew didn't work on, because it doesn't cover synthetic biology in plants. And so, the community that's doing these things does have some responsibility to start to think about how we regulate it to make it safe.
In the back, and then there's a Twitter question we'll come to after this one.
Hi, I'm Elizabeth Prescott from the State Department, and I wanted to ask, it's obvious there's, or at least from skimming the report, there's quite a bit of involvement from European DIY labs in the norm setting. I wanted to sort of understand a little bit about where around the world there might also be vibrant DIY communities, and to what degree you feel the norms that are described here apply in those contexts, and then to what degree there might be from whether FBI or other governmental agencies already involvement at the national level in non-European countries.
So the laws are very different in European countries around genetic engineering. So what we see is that the experiments are also very different because of that. For example, in La Palais, they don't do any, this is the Parisian group, they don't do any genetic engineering experiments whatsoever, but they do work with microorganisms. One of the microorganisms that they're working with produces a pigment, and they've decided they want to turn this pigment into an ink that you might use in your pen. Now, that's an interesting project. It doesn't touch on any of these major concerns. As far as I can tell, there are groups all over the world in various stages of organization. La Place, the French group, is an interesting one because they have Parisian backing. Basically, the Paris government has decided to hire 2 full-time employees for them and give them free space in downtown Paris, which is very nice for them.
It's really an innovation.
Yeah, I mean, it shows, at least to me, that there's faith in the French government that these groups can really produce innovative new projects, almost like an incubator or a biotech nursery. I mean, there's been talk about, well, Nathan and I have been talking about ways in which the program that we have with the FBI, which is basically a program about dialogue, how it could be exported internationally. There's been murmurings about it. I don't know how far it's gone.
Sure. So just to expand on what has happened, in 2012, the summer of 2012 in San Francisco, we invited not only representatives from the United States, but internationally, from 10 different groups internationally, to include folks from the UK, France, as we spoke about, Germany, the Netherlands, Indonesia, Singapore, to have a dialogue. So as I said before, we built this relationship between the FBI and the DIYbio community. We feel like it's a very fruitful relationship, and the motivation there was to expose an international audience of amateur biology members to that type of relationship, to show ways in which law enforcement and amateur biologists work together to benefit their situations. We were not projecting a model that they should adopt, because again, the circumstances in different countries vary. Relationships with different law enforcement or security defense ministries are very different in different countries. So we were laying out a model that we know works very well here to then have these other representatives take that back and assess whether or not that might be beneficial for them. Now, as a result of that, we have received multiple media inquiries from international entities asking a lot of these questions. You know, why are you engaging? And so with that allows us to not only talk about amateur biology on an international scale and talk about the good things that are happening here in the U.S., but also to sensitize them as to what it is we're trying, the best practices we're trying to share. So yes, what we look to share a lot of the things that we do, even to the normal course of what I do on a daily basis and what our organization does. We want to help other countries and other entities prepare themselves as well to be proactively addressing certain items. And so we're always in discussion about ways in which both the FBI and a law enforcement / security mission can be pushed forward to address any particular situations, as well as helping to socialize more of the concept, the the interactive concept, and also the good things that are being done in amateur biology.
And I'll just expand on that. So, as Dan mentioned, next year the iGEM competition is going to open up for the first time to DIYbio teams exclusively. GenSpace was an entry 2 years ago, but was affiliated with a college university. But so next year, DIY teams are actually going to be able to enter. And I actually think I sit on the biosafety committee for iGEM. That's another opportunity, particularly from an international outreach perspective, to sort of get a lot of these services and information to those teams, because they will be required to meet the biosafety standards and biosecurity standards that all the iGEM teams are required to meet. So it's another opportunity to sort of reach out to some of these other countries. I think the hope is that some of these teams may come from developing countries that may not have a university that was able to support a team. So I think that's another opportunity. I believe we tried to do this coding workshop in a couple of other places, and it was funding limited. We didn't have that much money to do it, and if I remember correctly, we reached out to the State Department to try to get some funding to do that, and there was some apprehension based on where some of the groups may be coming from. And so I think that's a hurdle when you start then mixing some of these DIY teams with countries that maybe, say, like Iran or some other countries that the U.S. from a government standpoint isn't on the best terms with. But that's a barrier. And so hopefully, with iGEM, it might be another entry point into sort of dispelling some of these this information to some of those teams.
I mean, I would add that most of these countries have really strict regulations around genetic engineering, and so if we do have European or international teams, probably the projects they're going to do don't involve any synthetic biology. Maybe they'll be computing or bioinformatics or something else, just because of the nature of the laws in their countries.
On the other hand, one of the DIY community members from Ireland was the first individual to get a genetically modified organism license to do work in what was essentially his home lab. And so it's the small enough government that he could pick up the phone and call the person who handles laboratory licensing for genetically modified organisms and say, look, I'm starting a small company. I'm a one-man show, and this company is going to be, run out of my house, basically, and I'm interested in working on low-cost open-source biotechnologies, and this is what I'm about, and was able to get a license, and paid the fee and got the license. So I don't know all of the different at least in Europe, all the different rules and regulations, but I would imagine that there would be pathways for these community labs to do something similar, but I don't know. And with the rest of the world, I don't really have a good perspective about Asia. I don't know if either of you do, but it seems like there at least that isn't apparent to me. There doesn't seem to be a lot of activity there. There is increasing activity in South America. There are groups setting up, and many of them are very early stage, more of the sort of special interest group of a meetup of having coffee and talking about ideas rather than actually having lab spaces. But so far, the most number of groups are in the United States and Europe, and I don't think there's any activity happening, to my knowledge, in Africa. At least that would be considered part of our community, or that we've had any discussions a little bit in Australia.
Do we have a question from Twitter?
The Sinberg Twitter account asks, what is needed to help the DIYbio community share and disseminate best practices for self-governance across diverse groups?
Yeah, money. I mean, yeah, it's always money, right? But it's not a lot of money, right? So a lot of these things that we've sort of put in place, in essence, the heavy lift has already been done. So the Ask a Biosafety Expert program, for instance, the web portal is built, and that only requires, in essence, limited funding to sort of keep that going and potentially expand it. I think, funding for sort of, potentially type like DIY conferences where you can, bring these people in together to actually sit around and talk to each other is another good way to disseminate a lot of this information. So I would say in order to sort of project out a lot of these best practices is that it requires some fundraising to sort of have, I think, very, in the essence, simple sort of meetings and other programs to get this information out there.
I also want to push back a little bit on the terminology best practices, because I think we have to be realistic, and that we're not talking about best labs and best practices. We're talking about sufficient practices to be safe. If you look at the types of devices and things that people are doing, it's not as if we have, luxurious options to do the best of everything. And, we don't serve caviar at lunch at community labs and those sorts of things. But, it's a terminology that I think has grown up that is kind of wrong, because this is the same conversation that I'm having also with the biosafety experts, is that they want to figure out what the absolute best practices are for every question that comes in, which I think is aspirational. But at the same time, I think there is a level of just sort of sufficient practices that is a good place to begin. Sufficient best practices, and just on the funding part, absolutely. A lot of these groups have been totally self-started, grassroots, pull yourself up by your bootstraps, including this Ask a Biosafety Expert, and that's all voluntary run. Everybody's contribution to that is totally unpaid and voluntary. And there could be equivalent or similar services that address all kinds of issues related to sufficient practices for setting up, community labs that are safe. There could be governance issues about, here are the basic resources to get up and to practice safely in doing molecular biology, or to adhere to local, state, federal regulations of what it means to set up a BSL-1 lab, that kind of thing. It's totally a funding, it's limited by funding. So anybody has good ideas about that, that's certainly something that would be.
Let me piggyback on what Jason said. It seems to me that there's sort of a formula for how community labs get generated. It starts with these meetup groups, like you were talking about in South America, people getting together, sharing an enthusiasm about the technology. Then usually there's some initial experiments, local, usually in someone's apartment or community space, where they're doing some of the most basic biotech experiments, like extracting DNA or protein purification. And when that core of people sort of come to a head and decide that they want to take their experiments to the next level, you get these small community labs. They literally go out and rent space and get members and start hosting classes and workshops and all that other stuff. So it seems to me that there is sort of a blueprint for how to do this. And I would argue that probably the most important thing to do would be to actually publish the blueprint, actually get maybe not best practices, but a set of practices around how to go from beers and DNA extraction to I have a community lab set up, and those would include practices for safety. They would include setting up a biosafety advisory board who could lead you through some of the thorny issues around what kinds of experiments can we do, to how do I sustain one of these spaces economically? How many workshops do I need to have to sustain my space? What kind of space should I rent? All these questions that you have to decide when you're actually out there searching for space. It just seems like we're at a ripe time where a number of community labs have gotten to a point where they're self-sufficient to some degree, and those practices could be shared. So if I were Sin Burke and I don't know, if I was sitting on some money under my mattress somewhere, that would be something I would support.
What strikes me most about this is that, and especially listening to you all, is that there are no analogs. We can make sort of generalized, connections. This is somewhat like the Apple startup, or this is some like Marie Curie and her lab, But in fact, and it's so important that you all are taking this seriously and under your wing and doing this, there are no analogs. And I think we have to start from that point. It goes back to your comment, there are no best practices.
Mm-hmm.
I mean, I don't want regulation at this point in time. I mean, I'm pretty much more on the side of science than anywhere, because I think it's the wrong place to be. However, because I do understand the science, and I was very concerned about the Kickstarter project with BioCurious, more from a scientific standpoint of what are the secondary fallouts here?
Mm-hmm.
What are the things that you're not going to be able to predict? I mean, if I look at ecological streams in how, for example, the blue butterfly was eliminated from the UK, in the end, it was the very innocent sort of concentration on rabbits. But the flow point from the butterflies to the rabbits was actually a bacteria, which was very unexpected, and it reminds me of Eric Chivian at the Harvard School of Public Health. And the one word I associate that with biology that has now burned itself into my brain, which is that it is irrational. And labs are highly rational. So that's where probably if I were to center a concern, it would be there. And to that place, I would say I would really like to see a kind of a government oversight panel. But I would draw people from CDC, because I see the greatest analog in this to CDC. I mean, they are both a primary research, discovery, and also safety, and they have a great deal of expertise in highly mutable organisms. I would do NAS, National Academy of Sciences, just because it's the best place for you to be right now. I mean, that's what's driving this, is curiosity, personal discovery, the top three in your why are you doing it. Mm-hmm. The rest all have monetary implications, which is also a pretty important reason for doing it.
Yep.
And that can act either as a constraint or a driver. I mean, with the Kickstarter campaign, it's now acting as a constraint, because people are saying, do we want that money in that quantity to flow to a project that we don't really have a good grip on? And I'll leave out Monsanto and patenting and all of that.
Right.
Let's say that's somewhere back there. But also would include NSF, because NSF has an interest in this, and they're very good at looking interdisciplinarily, which I think you probably need to, and not only physical sciences, but also social sciences, which goes back to the governance issue. Mm-hmm. And NIH, of course. But I think there are some places that need to be, and you're at the place now where you need to address it. And the fact that you don't have the funding to do it, I think if you go to the CDC, NAS, NSF, CADRA, and look at that internationally, I think you'll find the money. And not only that, but you'll find the sense of opportunity, like the Parisian group. And I know EU has this different take on bioengineering, and I have a tendency at this point in time, because we're not so terribly experienced, to be in their camp. So that's probably, and I guess that's more of a comment than a question, but I think it would be nice to see from you all, where you think you should go next. Because I think these components will probably help you get there, and it will also mitigate a lot of the fears, the myths. Terrorism, I don't see terrorism as part of this mix so much. But I do see unintended consequences when you get outside the lab, as a real touch point. it's like, well, where do you take it? We talk about human consequences, but there's also, I mean, what is it? Microorganisms are 1 billion times more numerous on the planet than there are stars in the universe. I mean, that gives you the potential and the understanding of how terribly creative, opportunistic, and irrational, are the opportunities, for little organisms to make big changes. And probably Craig Vender's group is a place that I would look for probably the greatest insight on that. I know they've done some work with the University of Illinois ecologists and microbiologists. So it's a kind of a segue, to that kind of other implication, other world.
So one reaction to that is part of, I think you said a lot of really interesting things there. And thanks for your thoughts on that. I think part of the frustration with the terrorism discussion is that it is, in my opinion, like the biggest time suck and resource suck when we actually need to be having more interesting and practical questions than that. My personal opinion, these are all my personal opinions. But, not that, watching for terrorism is important, but I think that in my opinion, and based on the things that I see people doing and wanting to do, actually working on some of these more practical questions related to, environmental release and appropriate management of these technologies, and having supportive intellectual work done here, I think is really an important thing. And it's just been frustrating in some sense to take a 4-year detour and pause on having every other conversation because we couldn't get out of the terrorism box for years. So much so that I know me personally, I unplugged my phone not really, but I took no calls from journalists for 2 years because there wasn't anything that I could say. It was like the example with the Baltimore group inviting the FBI for a meet and greet, and the headline is FBI takes interest in local group and totally turns it around on us again, or turned around on Baltimore group. That's really frustrating. Mm-hmm.
Scientific, international scientific or oversight group, keep it very small, would help you to mitigate those fears and tamp it down.
Yeah.
Because right now it looks like this wild grassroots movement.
Yeah.
And it just is, that allows it to be characterized this way in the media.
Mm-hmm.
And that's one of the things I think IPCC does well now, is that it has enough credibility and oversight and good scientific voices to defend its rather complicated, what's invisible to most people, scientific glacier.
Yeah.
It's like what the media picks up is the little tip of the glacier.
Yeah.
But you also need to have somebody in some small group to talk about that, huge submerged piece. And I just think it would take that off of you.
Mm-hmm.
And put it in a place where people are more receptive. So, a thought.
Yeah, I'd just like to sort of, I kind of agree with almost everything you said. I want to use the Glowing Plants project sort of as a discussion point because, we've been working together we've tried to figure it out probably 3 or 4 years now, trying to sort of set up these types of programs for the community to be able to operate safely. And if you look at the codes of conduct, the Ask a Biosafety Expert program, the fact that most of the community labs have biosafety councils that have very high level quality people on them, I argue that the DIY community is actually ahead of the game in terms of a lot of these questions. And if you look at the Glowing Plants project, I would argue that all the systems that the DIY community put in place worked. So BioCurious did not allow that project to happen within BioCurious because there was going to be an environmental release of the seeds. So now there's a double-edged sword there, right? So now they had to go out and find another space to do that. But I think from the DIY community's perspective, they've in essence followed the codes of conduct that they developed themselves organically within the community, which one of them was, there will be no environmental release of our projects. And so I would argue that those systems, that system for that particular project, from BioCurious' standpoint, the DIY community standpoint, worked from a governance standpoint. Are there ecological issues associated with sending out those seeds? Absolutely, right? And so are they going to be evaluated? That now becomes a governance question, right? It's sort of in essence outside of the DIY community's purview from a governance standpoint. Now that project, they are open to looking at those issues, which I think is a positive. And I think that also comes from the DIYbio ethics about the work that they're doing. And so I think the systems that have been in place up until now, I think have been working. Do they need to constantly be evaluated as the community becomes more advanced? Absolutely. But I think, what's happening up until this point, I think has been working. And I've argued the DIY community is actually far beyond the general SynBio community in terms of a lot of these issues because they've addressed them early, or have at least attempted to address them early.
Right. So right. So right. Right. It didn't become a part of the media conversation. And I think they wanted it not to be part of the media conversation, both sides. I don't think BioCurious wanted to be associated with it. And I think they quietly disassociated themselves. I think it's also interesting to note that because the community is so communicative with itself, it's so well networked, that when that happened at BioCurious, there was a whole debate about ecology in other labs. And so one result was that one of the labs brought an ecologist to come to talk about environmental release and actually teach an entire class on what happens when an organism gets released and what we can know and what we can't know, a year, 2 years, 5 years after such an organism goes into the environment. So to me, that says that the community's thinking about these things. I don't think there's consensus among anyone in the community about what we should do or what they should do in terms of that. But I think there's a clear sense, at least in the community labs, that releasing an engineered organism is just not on the cards. One of the things that a lot of people are working on are kits, right? So kits of genetically modified organisms where maybe you paint images using the GMO, right? So we saw the mosaic in the slideshow. My sense is that they're not jumping ahead of the scientific community. They're just sort of using the technology that's out there, using technology that's been around for dozens of years, and presenting it in a new way. So I think you're right. We need to have these conversations. I think it should probably start now, and what you're saying makes total sense. But at the same time, I think we need to be. I think we should realize that actually the community has begun discussions on this already. It's organically.
Yeah, there's no, it's not a sort of consensus-driven community, or there is no, That's
part of the scare factor.
Right. Yeah. Yeah. But at the same time, on the other hand, there are organizations that have leases and clients and neighbors and want to be viewed as a community resource and not, a threat to your neighbors kind of thing. And I think that is one of the most important aspects of DIYbio not to forget.
Right. They have to deal with their community, not and their neighbors essentially, and the local fire department and the police and everyone, in, you know, and the nosy neighbor downstairs as well. So they're very cognizant of the fact that what they're doing could be seen as dangerous, whether or not they are doing something dangerous. And a lot of that concern and the concern for, hey, we're an education institution, and we want you to come and see what we're doing. We want you to participate and see what we're doing is not dangerous. Adds a lot to that argument, and I think mitigates a lot of the risk. I think at this point, these groups have formed organically, and so that wasn't ever an option. I think if Stanford came to BioCurious and said, "Hey, we're going to give you money and free space for you guys to come in at any hour that you want, teach classes about anything you want, and work on anything that you want, given that it's biosafety level 1," I think they would jump at the opportunity. But I've never seen an offer like that on the table. Now, Paris got that offer, right? With the Parisian government, right? And of course, they jumped at it. Who would turn that down? Interestingly, I talked to Thomas, who's one of the directors of that lab. He said, well, for the next 3 years, we'll take the funding, but going on out, we really want to be self-sufficient, which is an interesting decision on their part. GenSpace is another example. They're looking to hire staff, and to do that, they need to get funding. They need to apply for grants, and they need board members who basically can bring in money, and so that they're changing their model as well. So we might just see that. We don't know. Time will tell.
Any other questions? All right. Well, I really want to thank Dan and Jason and Ted on the phone, if you're still there, for coming and presenting the report, and thank everyone here and on the web for watching. So thank you very much.
Thank you.